Hello everyone. So in this session we are starting with the prions and likens and viroids. Before moving to this viroids and prions and likens, we'll have a glance on the structure which I have drawn on the diagram on the board. So the diagrams are, this diagram is known as tobacco mosaic virus and this is known as bacteriophages. So these two are incomplete diagrams. Why so? Because I have not labeled these diagrams. Let us study the few description of the TMV and then the bacteriophages. Before this, we have completed the viruses. So what are the two types of viruses? Three types. One is plant virus, animal virus and bacterial virus, and what type of genetic material it included, how it infect to other host cell, outside the host cell, how the virus act. So all these things, and who discovered these viruses, and what is contagium vivum fluidum. So all these things we have completed in the last session itself. So we'll move on to the next concept, that is structure of TMV, tobacco. Let me write the label headline first, later I'll draw I'll complete the diagram and then I'll label this. So this is tobacco mosaic virus. In short, we used to call this as TMV. So the name itself saying this virus is going to infect only to tobacco plant. The leaf of tobacco plant, it first infect. The virus infect to the leaf of the tobacco plant. When the leaf it begin to arise, then only the virus start infecting to this leaves. How we can consider the tobacco plant or this leaf is infected by this virus? We can see the mosaic patchy like patches on the surface of the leaf, so that we can conclude yes, the plant has infected by some virus. So this is the first stage or first thing what we have to understand in the tobacco plant. Only the leaf get infect, and then how this leaf will become after getting patchy on the surface of the leaf. All this surface it become yellow, and after that we can see green patches on the leaf. Primarily what we saw only some mosaic like patches on the leaf when the leaf was in the preliminary stage or in the leaflet. When it matures, it lacks or it fails to produce the chlorophyll. It fails to synthesize the chlorophyll. When the chlorophyll is not there, it cannot synthesize the food material. It cannot spread its color into green. So after that we can only see a green patches. Green patches indicates the chlorophyll. Only in that patches chlorophyll will be present. Okay, so when the scientists they discovered this virus, it looked like that. So the virus consists of a nucleic acid. As I told you in the last session itself, the plant virus consists of RNA. The animal virus consists of RNA or DNA. Okay, so any of the virus consists both the RNA and DNA. Virus, either it involve the RNA or it involve the DNA. Okay, so So we are speaking about the plant virus here. So what we have to consider, so if I am writing this as nucleic acid, what you have to remember, what you have to learn this, what type of nucleic acid is present in the tobacco mosaic virus. Here it consists of RNA. RNA in sense ribonucleic acid. So this is the one description, one character what we are giving to the tobacco mosaic virus. Now we can able to see some other structures in the tobacco mosaic virus. What actually it is, those who have studied this textbook, they can answer this. So what is that? It is nothing but a protein coat. Protein coat in sense it is a capsid. So the nucleic acid is protected by a protein coat known as capsid, and this capsid made of number of subunits known as capsomeres. So we have given three characters to this diagram. So one is, it consists of single stranded RNA, which is a genetic material, which is an infectious agent, which is going to infect to the host plant or host cell. 1 point. So let me complete this diagram. So here you have to make some circles. One more question arises. Maybe you are thinking how to draw this diagram. Already I have drawn this diagram on the board, so you'll get confused. No, so no problem. I'll teach you how to start this diagram. Okay. First, what you have to write, what you have to draw. When they gave this diagram in the examination, how to start this diagram. So this diagram includes nucleic acid, right? Just draw the nucleic acid first. So this is the nucleic acid or RNA, what I have drawn here. So just make it one more line so that it looks like that, and draw the outline. Outline in sense, just do it like this. Later you have to erase this, that the TMV doesn't include any of the borders and all. Only to make your diagram perfect, just draw the outline. Alright. So now what you have to write, you have to show the protein coat. Protein coat is nothing but a capsid. Capsid is consists of number of subunit. Number of, this is one subunit, 2, 3, 4, number of subunits are there. No, how to show that subunit? For that, just make the outlines here like this. Remember, don't forget to erase these outlines. Okay, so now what you have to write? You have to write the subunits or capsomeres. Got it? So this is now the TMV diagram is ready now. Now just erase the outlines and if any of the subunit got erased, just rewrite it. Okay, now coming to the RNA, what you have to show here, you have to show the circles, what I am drawing here, no. So that's all about the structure of TMP, drawing in a easiest manner. Now, RNA, capsid and capsomere. So, maybe the chapter is for only 2 marks. No, weightage is only 2 marks. So, only if they ask, they'll ask only to draw the diagram. They won't ask you to describe the structure. If they ask, no problem. Whatever the diagram, whatever the labels are there, no, just write it as it is. It consists of single stranded nucleic acid, that is RNA, the genetic material. So this is the genetic, this is also known as genetic material. This genetic material is covered by a protein coat known as capsid. The capsid is made of number of subunits known as capsomeres. So that's all about the TMV, what we are talking about this diagram, tobacco mosaic virus. So we'll move on to the next diagram. The next diagram consists of, it has like one diamond shaped structure is there, some tail like structures are there. So what does it mean? It is so simple. If they ask you to draw the bacteriophage, the name of this diagram is bacteriophages. Phage, bacteriophages, not S, just bacteriophage. Okay, so we'll describe the structure of bacteriophage now. So let me tell you about the bacteriophage. The bacteriophage is nothing but a virus which is infecting to the bacteria. It is known as bacteriophage. So this is a virus. It is going to infect only to the bacteria, not any of the plant or animal material. So those kind of viruses are known as bacteriophage. Okay, so now we are going to learn the structure of bacteriophage. It includes, no doubt, genetic material or nucleic acid in the form of DNA. But in this diagram, we studied the genetic material is in the form of RNA. But here, the genetic material is DNA. And this is known as head. Okay, so the head is at the anterior position. We can see a head. Inside this, the genetic material will be present, and this is known as collar, and this is the sheath. And so you can see 3 pairs of some spikes are present at the posterior end of the bacteriophage. So what is that? It is nothing but tail fibers. Tail fibers. Okay, so how many tail fibers are present? 1, 2, 3, 4, 5, 6. There are 6 tail fibers are present, which is connected to the end plate. So this is the end plate of bacteriophage. Okay, so inside to the head there will be the DNA will be present. Okay, and a collar and the middle piece or sheath, and here will be the end plate. To the end plate, 6 spikes got connected. Spikes or tail fibers are present. let me show you how to draw this diagram. So don't worry and don't get confused and don't write the wrong diagrams, just write the right diagrams or correct diagrams. First what you have to draw, you have to start the bacteriophage. If you want to draw the bacteriophage, you have to take or you have to start with the head part. So just draw the outline first so that you can complete your diagram. So this is the outline of the head and some lines over there. Okay, now some more lines. Okay, and like this and like this. Got it? And then what you have to draw next? You have to draw the collar, and next to that a spirally coiled sheath like this. And then how to draw the end plate now? Just look at this. When you start drawing the end plate, take your hand from the left side of this sheet and draw like that and like this. Yes, that's all about the end plate. Now take the fibers. How to draw the fibers? One fiber should come behind to this. So one fiber, one more fiber coming from this end and one more fiber, and at this end, this side, one fiber is coming from the behind to this bacteriophage. One is from this end and one more is from this end. So make it proper. I have just drawn as it is. When you draw properly, you will get a correct shape of the bacteriophage. Okay, so that's all there in the diagram of bacteriophage. Okay, so this will become easier for you to draw. Now, so this end plate, whenever it want to infect the bacteria, this bacteriophage, if it want to infect the bacteriophage, first it will pierce the bacteria from this end plate. First it will sit on the bacteria, the tail fibers, it will hold the bacteria, and then these end plates, it will penetrate to the bacteria, and then from this side the genetic material passes and enters inside the bacteria, and when the bacteriophage, when it releases the genetic material inside the bacteria, there the genetic material increases its number. Increases its number means it will get multiply by using the bacteria proteins and then bacteria host cell. So bacteria means host cell genetic material. By utilizing the host cell or bacteria genetic material, it will multiply its number. Okay, so that's all about the diagram of bacteriophage. So we'll move on to the next concept, that is nothing but viroid, prion and lichen. So we'll start with the viroids. The viroids, it was first discovered by T.A. Diener in 1971. T.A. Diener was the one who discovered these viroids. Why the name came viroids? After the viruses, why it is necessary to read to understand the viroids? Because the viroids are smaller than viruses. Let me write the characteristic feature of viroids here. So this is considered, this is the first character. So you have to remember this. T.A. Diener was the one who discovered the viroids, and the next character is these are smaller than virus, and these viroids are, it doesn't have any protein coat. It lacks the protein coat. Protein coat in sense, in the previous diagram we studied the TMV, right? The TMV is the one, is the virus which has the protection in the form of capsid. So that capsid is nothing but protein coat. So that kind of protein coat is absent in the viroids. Then what else? Only it has a free RNA that will be moving randomly from here to there. Whenever it finds the host cell, it will just enter and it causes the disease. It has only free RNA. Free RNA, why I have written here free RNA? Because it doesn't have any protein coat, only RNA is present. This RNA has a low molecular weight. So this is also one character you have to remember here. So these viroids infect to potato plant. When this viroids infect the potato plant, it causes diseases. The disease name is potato spindle fiber disease. Spindle fiber disease. If they ask you to write the disease name infected by viroid, what you will write? You have to write potato spindle fiber disease caused by viroids to the potato plant. Okay. So the next is, this is only smaller than virus as I told you, and it doesn't have any protein coat. It has only an RNA which will be freely moving. When you want to infect the organism, the RNA itself enter inside the host cell, and there it causes diseases. The RNA will be of a low molecular weight. This viroids infect the potato plant. This potato plant, when it get infected by the viroids, it will be causing from the disease known as potato spindle fiber disease. Okay, so we'll move on to the next concept after the viroids. So just let me tell you one more thing here. Thus, these are smaller than viruses. Hence, it is known as viroids. Just add one line here. Hence, it is known as viroids. Okay. So, that is the one thing you have to remember. After the viroids, The next concept is we are going to study about one more microorganism. The name of this microorganism is prions. A newly invented medicinal field. Some of the scientists, they discovered some protein-like structure. Protein-like structure it is. It doesn't have any of the genetic material, any of the RNA or DNA. Some protein-like structures are going to infect to the eye tissue, brain and spinal cord. To whom it is causing? To animals and human beings. In animals like cow, sheep and humans, if it enter inside the body, the prions means what? It is not an RNA or not a DNA. Then what actually it is? It is a protein like structure, okay. Whenever it enters inside the human being or sheep or cow, it directly infect or it directly infect to the brain, spinal cord or eye tissue. Okay, remember this word. Alright, so what will do this prions? It will alter the structure of proteins. What these prions are doing? These will alter the structure of proteins which is present in human being or in the sheep or cow. Okay, second thing, to whom it is causing? It is causing diseases to animals and humans. Mostly, recently, the cow. Let me write the disease name of that disease name infected by this kind of microorganism to the cow. So the disease name is BSE. What does it mean? It means bovine spongiform bovine spongiform encephalopathy. So this is one name of the disease caused to the cow. When the cow is suffering from this kind of disease, this disease commonly known as mad cow disease. Mad cow disease. Okay, so what does it mean? So the prion, whenever infects to the cow will be suffering from BSE type of diseases. Commonly it is known as mad cow disease. So the symptom will be, it will be acting like mad or crazy. It will be acting like a crazy cow or mad cow. By seeing the symptom, we can make sure the cow is suffering from this kind of disease caused by prion. Okay, so one more disease is there, which is usually seen in humans because of this type of microorganism. Creutzfeldt-Jakob disease. Jakob disease. The name of the disease is Creutzfeldt-Jakob disease. CR Jakob disease. In short, CR Jakob disease. Why they have given that type of a name to those disease? Because he was the one who discovered this type of diseases. That's why they have given the name. Okay, so the human, they too can suffer from the diseases caused by the microorganism known as prion. How it will enter inside the human body? So maybe these proteins, or maybe in the NCERT book they have called the prions as agents. These agents, through the meat or through the saliva or through any of the media, if it enter inside the human body, firstly it will infect the brain tissue or to the brain, spinal cord or eye tissue. If it get infected, just imagine if the brain is infecting by some other kind of microorganism, how will be the condition? Just imagine by yourself, and please make sure you are doing or you are leading a hygienic life. So because we cannot say which type of microorganism is there in the environment. If it enter inside the body, it is going to cause an uncurable diseases. So please be careful. So at last we are going to learn the last concept of this chapter, that is likens. So likens means, maybe you have seen on the tree trunks, on the rock surfaces, or on the either on the tree trunk or on the rock surfaces, the lichens usually grow. How it looks? Somewhat it looks like this. And it doesn't have any color. It appears like gray color or brown color or cream color. I'll show you when you come to the college. I'll show you how the lichens look. Okay, so what actually the lichens is? What is there in the lichen? Lichen consists of two organisms. One is algae and one more is fungi. Yes, inside this lichen, two type of microorganism used to live inside this lichen. So how they both live together inside this lichen? Lichens means, so it is not like a matter of either algae or either fungi. So this is the matter of symbiosis, a mutual understanding between algae and fungi. They both, so this is the matter which where they both are going to live inside the lichens. Okay, so the algal component is phycobiont and fungal component is mycobiont. How they are going to live inside in the lichen? It is not going to harm to the algae, or the algae, if it is not getting disturbed from the fungi. So if the question arises inside your mind, just don't worry about that. They are going to live by means of mutual understanding or by means of symbiotic association. What the algae is doing to fungi, the algae is going to manufacture the food and it will supply to the fungi. In the reverse or in the reward, what the fungi is going to give to the algae, the fungi is giving shelter and it is going to absorb the mineral nutrient from the surface and it is supplying to the algae. So this is how they both going to understand with each other and it is going to live with each other. This is known as symbiotic association. And the component of algae is phycobiont, fungi component is mycobiont. And one more thing to say, the lichens are the main pollution inhibitor. They do not grow in the polluted area. Pollution, so what does it mean? The lichens are a main pollution indicators. Pollution indicators means wherever the pollution is present, the lichens are not going to grow. If the lichens is present, that area is free from pollution. So that's all about this chapter. So after this chapter, what I used to give? I used to give the assignment, right? So just write down few questions related to the chapter number 2, biological classification, and complete the assignment, submit it to me as soon as possible. So primarily I'll give in the first main I'll give 1 mark question and then 2 mark, 3 mark, 5 mark. Okay, so first question is, what is biological classification? What is biological classification? Second question, give an example for unicellular organisms. Name the third question. Next question, name the 5 kingdoms of living organisms. Name the 5 kingdoms of living organisms. Who proposed 5 kingdom classification? What are monerans? Give example. What are monerans? Monerans means prokaryotic cell natured organisms are known as monerans. Example, bacteria. Why archaebacteria can also live in extreme conditions? Why archaebacteria can also live in extreme condition? because it has a cell wall composition, a different cell wall composition compared to eubacteria. So, because of this nature, they can survive in extreme conditions. What are halophiles? What are thermoacidophiles? Thermoacidophiles. What are methanogens? What are cyanobacteria? Cyanobacteria. Cyanobacteria. Give an example for cyanobacteria. What are heterocysts? Heterocysts. Heterocysts. Write one significance of heterocyst. What are chemosynthetic autotrophic bacteria? Chemosynthetic autotrophic bacteria. It means bacteria which is going to oxidize the inorganic substances like nitrate, nitrite, ammonia. So, by oxidizing all these, they can prepare their own food. They are going to live independently by preparing their own food and by oxidizing inorganic substances. That's why they are known as chemosynthetic bacteria. Next question, what are mycoplasmas? Mycoplasmas are smallest living organisms without cell walls. So, without cell walls, and they can survive without oxygen. So, it is known as these type of microorganisms are known as mycoplasmas. What are planktons? Planktons means which is going to float on the water surface. Those type of organisms are known as planktons. Name the chemical component of cell wall of chrysophytes. Chrysophytes. The walls of chrysophytes, so this is the question, okay. The walls of chrysophytes, in bracket if you want you can write diatoms. Name the chemical component. The walls of chrysophytes are indestructible. Why? Indestructible. Why? What is diatomaceous earth? What are chief producers of oceans? What are dinoflagellates? What are euglenoids? What are slime molds? Name the disease caused by trypanosoma. Give one example for sporozoan. Sporozoan means the answer is plasmodium. Okay. Name unicellular filamentous fungi. Unicellular filamentous fungi. Which fungi act as source of antibiotics? Name the disease caused by Puccinia. Puccinia. What are hyphae? Hyphae. Hyphae. What are cenocytic hyphae? Coenocytic. Cenocytic hyphae. Name the cell wall component of fungi. What are saprophytic fungi? What are parasitic fungi? What are mycorrhiza? Mycorrhiza. What is plasmogamy? What are fruiting bodies? Fruiting bodies. What is karyogamy? What is mycelium? mycelium. Mycelium. What is zygospore? What are isogamous gamete? What are basidiocarps? Basidiocarps. Basidiocarps. What are conidia? Give an example for insectivorous plant and parasitic plant. Insectivorous plant and parasitic plant. Insectivorous plant like bladderwort, Venus, flytrap. Parasitic plant like cuscuta. These are the answer for the question. What are the two distinct phases of life cycles of plants? What is alternation of generation? What is capsid? Why viruses are called obligate parasite? Name any two diseases caused by virus. What are viroids? What are likens? What is phycobiont? What is mycobiont? What are the two components of viruses? Two components of viruses like genetic material, DNA or RNA. Second one is protein coat. Okay. Now, question carrying 2 mark. Second main, classify the bacteria based on the shape, SHAPE, shape. List out any 4 properties of cyanobacteria. Cyanobacteria. Mention the methods of reproduction in bacteria. Like if the answer will be fission, by spore formation, transduction, conjugation. Name the flagellated protozoan and ciliated protozoan. Flagellated protozoan is trypanosoma, ciliated protozoan is paramecium. Write briefly about lichens. Draw a neat label diagram of bacteriophage and draw a label diagram of TMV. Now, question carrying 3 mark. 3 mark or 5 mark. Now just write like this. Instead of 3 mark, you just write 5 mark question. Answer the questions for 5 mark. Write the salient features of kingdom Monera. Write the salient features of kingdom Monera. Write the characteristic features of kingdom Protista. Write the general characters of kingdom Fungi. Write the characteristic features of kingdom Plantae. List the characters of viruses. So just complete this and show it to me, and if you have any doubts, please ask me. Thank you.